Welcome everybody to the first and hopefully last ADRC virtual meeting. I hope next time we get to see each other in person. But we have a full packed schedule today, so I'm not going to do too many introductions here, other than to say I hope everyone is doing well and staying safe and not going crazy being at home. So thank you for joining us at this virtual meeting. You've entered the meeting with a muted microphone as an audience member. We'll be using the raised hand function. It's on the lower left-hand corner of your open participant box menu. Please raise your virtual hand at this time to demonstrate you can hear us at the located function. Okay, so primarily depending on the remaining time for questions and answers, we're going to be gathering your questions in the question box. Please find the question and answer selection on your menu and type ADRC. Cerise will be managing your questions, announcing the questioner's name and reading it aloud for the presenter to answer and hear the audience. So our schedule today is really jam-packed full. So we can try to get through all these really important things today. I'm not gonna spend too much time talking now. And also I'm gonna really need to have people stay on time. So we're gonna be ending presentations that reach the 1 minute mark towards their scheduled time to try to allow for questions or a concluding remark. And we really hope that that the presenters have allowed some time and questions and answers. I know there's going to be a lot of questions today, given everything that's going on. So without further ado, I want to welcome Dr. Richard Hodes, who's the director, as you know, of the National Institute on Aging, for the first presentation. Welcome.
Thank you, Julie. So I will try to convey information efficiently, leaving time, as you suggest, for discussions, questions. Next slide, please. So an obvious opening is to discuss what we understand to be the profound consequences of the COVID-19 crisis for all of you. And I just wanted to briefly show this slide with information that we can share with you as the whole slide presentation, just to indicate that we're spending a great deal of effort trying to be responsive to information, questions you may have. Particular here is a funding opportunity announcement for administrative supplements, revisions, and emergency revisions to entertain under these extraordinary circumstances. And there'll be many of you interested. Staff will be happy to follow up with specific, but I wanted to introduce that as the unavoidable context of this meeting we're in now. Next slide. And next, so as usual, we're talking about updates on appropriations. And back before the crisis, we had another good year in appropriations across all of NIH, the overall budget of which went up to nearly $42 billion. For NIA, this was an increase that took the NIA appropriation a little over $3.5 billion. Again, an additional set aside for AD and ADR related research of $350 million, which brings the total allocation NIH-wide to some $2.8 billion. We all appreciate just a remarkable increase, but noting as well the increased NIA was not limited to only Alzheimer's related research, and so benefits all of the aging research that we support. Next slide. This is to show just the trajectory of increase in total dollars that brought us to the NIA total of $3.5 billion that I've mentioned. The top line, the current dollars, the bottom corrected for biomedical inflation, but clearly a remarkable increase, large measure of which was in Alzheimer's related dementias research. In the next slide. This is to show you year upon year what's happened, and the most profound changes beginning in 2016, which, as you can see, began the process of 350, 400, 414, 425, and $350 million increased appropriations, reflecting the national importance and priority for research in this area, as well, I have to say, the work of all of you, which allowed us to plan, propose, and demonstrate the fruits of research that are being carried out in pursuit of the goals of the national plan to arrive at effective interventions at earliest possible date. Next slide. In terms of pay lines, we live at NIA with very different pay lines for AD, ADRD, and for other so-called general research. Our pay lines for general research, under $500,000 direct costs. For base applications, 11th percentile, with additional advantage given to new and early stage investigators. In contrast, on the right, you can see for AD and ADRD, as for the previous year, a 28th percentile, and again, giving still further advantage to new and early stage investigators in an effort to recruit new people into the field. Next. For other mechanisms, program projects, other NIA research, career awards, you can see once again the pay lines maintained at levels for AD and ADRD, which gratifyingly allow us to fund a large measure of meritorious reviewed applications. Next. As you will probably be aware, we've made an effort with the appropriations which come to NIA to nonetheless serve the task most efficiently by supporting research across other institutes at NIH. And what we've done now in FY18, FY19, and ongoing in FY20 is to invite other institutes, all other institutes, to request AD/ADRD related supplements to grants from those institutes that are not themselves Alzheimer's and related dementias. The effort is therefore to recruit people with their own talents and perspectives into the field. Remarkably effective, as you can see, for FY19. These are the numbers of supplements given for each of the many institutes. NINDS, the largest at 41, but NHLBI not far behind. And you can see essentially all the institutes at NIH having come forward with some remarkably innovative and unique proposals for supplements that are AD and ADRD related. Next slide. In terms of the success rates this means for our grants over time, to show you that we've plateaued at around the 29th percentile success rate for those of you who understand the significance of success rate as opposed to pay line. They happen to be very close in this case. For NIA, now above the NIH average in large measure due to these recent increases. Next slide. I just want to make note of the SBIR STTR program, which is a set-aside legislated that is increased in its percentage and increased because our total appropriation increases. So the SBIR STTR program at NIA in this fiscal year is at nearly $120 million, and it allows us to encourage partnerships with small business directed towards the goals of the program, and encourage any of you who have any thoughts, who had not had previous thoughts, but are curious, to contact our office for help in assisting you to translate the research interests into these sort of translational opportunities as well. Next. So some updates, as we and the public are very interested in the number of intervention and prevention trials that are ongoing, focused at Alzheimer's and related dementias. This is one of the updated summaries that we present routinely. It totals about 230 initiatives and trials. They begin with the early stage clinical drug development that you can see, late stage, non-pharmacologic, therapy for symptoms, as well as a number of now interventions for care and caregiving. Among the features which I'll point out, if you look at the early stage clinical trials in particular, that of the 39 currently being supported, 9 are amyloid and 30 are to a diversity, an increasing diversity of other targets. So even as we pursue important opportunities around amyloid interventions, and they do predominate in our late phase trials, the next generation that's coming from discovery and moving into clinical trials is far more diverse, as is the set of non-pharmacologic and combined interventions. Next slide. So in order to achieve this transition as seamlessly as we can make it from early identification of potential targets through their validation, all the way through to drug approval, we've put into place infrastructure in which many of you have participated, in which many of the centers play an important role. And they run from AMP-AD to identify targets, potential novel targets, to the newest centers program just launched last October, Treat AD, and the next slide sort of highlights that to explain its novelty. And this in turn is intended to feed into the model systems and ultimately into our infrastructure for clinical trials themselves. Next slide, a little bit more about these newest centers. 2 new centers, which over their 5-year span will be appropriated some $73 million, designed to diversify still further our drug development pipeline and provide infrastructure for those interested in taking their candidates through to the drugs. Next. There's new a NIA landing page I'd point you to. The centers have been very much at the positive and cutting edge of aggressive data sharing, which we appreciate and applaud. This is a resource identifying all the data sharing policies as well as resources that exist that are relevant to the work around NIA and in particular to AD and ADRD research. Next slide. I did want to update you on recent events this now past month. As many of you will remember, we carried out a couple of years ago an initiative in which we asked AHRQ, evidence-based practice centers, together with national academies, to evaluate a question. That question was what the level of evidence was for effective interventions to slow or prevent Alzheimer's or cognitive decline. Just want to comment to you now on another similar intervention we've carried out to ask, in the face of the important emphasis now on care and caregiving, what the level of evidence is for the effectiveness of programs that have been reported and published. And it's in two parts. The first part, next slide, was to ask the Agency for Healthcare Research and Quality, AHRQ, to do an evidence-based practice review on all the published work in the field. Next. And then the second will be the national academies taking the results of that survey from AHRQ and rendering further through a panel of experts in their identification of levels of evidence and, importantly, research gaps. Next. The status of it, and the next slide, please, was that in just a week ago, really, the AHRQ presented to the committee of the National Academy the draft of its report. And the next slide. Next slide, please. And that is available for any of you to take a look at. Basically, it indicated very little in the way of high level of evidence for effective interventions and pointed us to the next phases of needed research. As you know, for all of our research, we track our implementation through goals that feed into our annual bypass budget as well. Just invite you here to understand that we track through this 150 some odd implementation milestones over time at a website that's open to all of you to see as we understand the progress being made and reciprocally the research gaps that need further attention. The next slide. Just to show you how things function, if you look at our total sets of milestones in this year, this past year, the number that are completed were 26, 69 in progress, 19 to be initiated. And we do this to track our accomplishments and successfully meeting milestones as we work. It's important for us to make sure that the research is bearing the fruits that it's intended and that we have plans for the research needed to address our gaps. It also is very effective in communicating to audiences, including all of you, advocates, congressional audiences, the way in which we are, in a very objective way, monitoring the considerable progress that has been made and is being made. Next slide. And finally, just in terms of keeping informed, we try to maintain a lot of this information available on the web, active funding opportunities, approved concepts, which mean they've been through our council and they may be developed into funding announcements that would subsequently be provided to the public, and invite you as well to subscribe to our blog so in a regular way you can be informed of updates as they occur. Thank you. And I hope this leaves time for any discussions now or later in the morning.
Thank you so much, Dr. Hodes. Are there any questions? I don't see anything typed into the question answer. If anybody has questions, please feel free anytime to type something in and we will respond. But thank you again, Dr. Hodes. And now I want to introduce Dr. Masliah, the Director of the Division of Neuroscience in the NIA.
Good morning, everybody, and great you're joining us. And again, just to send my regards to everybody and hope that all of you are doing great and hope to see you in person soon. So next slide, please. So along the lines of what Richard was mentioning, we continue to make a tremendous progress in advancing the milestones, and there are a number of areas where we're focusing, including greater integration among basic preclinical and clinical programs, early detection and diagnosis, including biomarkers, the national strategy to enhance recruitment for AD clinical studies that we have talked to you about that in the past. And we just heard also how we are enhancing the clinical trials pipeline and with more shots on goal. And all of this is through an integrative approach and trying to advance as much as possible data sharing, for which of course the centers play a central role. And here I just want to take the opportunity to introduce a new member to NIA and to our Division of Neuroscience, Jenny Larkin. She's coming from NIDDK. She has experience in genetics and machine learning, harmonization, and she has a background also in neuroscience, and she'll join as deputy director, but also be part of the genetics and populations team. But she'll be working with Susanna and Lori and the rest of the team in the data sharing, harmonization, and so on. And I hope to be able to introduce her in person next time we meet. So the next slide, please. So as I was saying, the Alzheimer's centers really play a central role in these data sharing efforts. They are really the glue connecting with the AMP-AD target discovery, MOVE, resilience, and neuropsychiatric symptoms, but also connecting with the epidemiology and cohorts with ADNI and ADSP, and advancing other programs like, as Richard mentioned, Model AD and Treatment AD and ACTC and ABCDF. So really, the Alzheimer's centers play a central role at many levels in these data sharing biosamples. The next one, please. Now, this is new data from iDROP. iDROP has been recently upgraded and modernized, and all the data from 2019 fiscal year being entered from not only NIA, but also from multiple sources. And again, I invite you to visit. Lots of very exciting tools and data you can look at, but along the lines of what Richard was saying, and you can see the trends, how they've been changing over the years. In blue is amyloid as a target and the AD drug discovery and preclinical drug development projects, and how the A-beta related projects have been decreasing, while other areas like tau, inflammation, receptor, cell death, ApoE, proteostasis, all of those have been increasing over the years. So we really have a very diversified portfolio in the drug discovery and preclinical drug development areas. And as a reflection of this, and the next one, please, if you look at the molecular and cell mechanisms, again, a very similar trend. While in the previous years, maybe 5 or earlier years ago, we had a preponderance of A-beta and tau research funded. In the last few years, we have a tremendous diversification with circuits and synapses also increasing, inflammation increasing as a research, ApoE, etc. Really phenomenal diversification thanks to the influx of funding and the diversity of new programs that are being developed. And along those lines, in the next slide, please, I think also a tremendous advance that we have seen in the last year or two have been this greater understanding of Alzheimer's disease in the context of aging. And I know that we have put a tremendous effort and time in understanding Alzheimer's disease in the context of a proteinopathy, but there is also now tremendous efforts on trying to understand how the proteinopathy interfaces with mechanisms of aging like DNA damage, cell senescence, immune surveillance, and so on, and how the two pathways actually play a role in the neurodegenerative process in Alzheimer's. And as a reflection of that, if we go to the next slide, please. The previous one, go. This last fiscal year and this year, we have had a number of workshops and initiatives along these lines. Just a few weeks ago, a couple of weeks ago, we organized a meeting on a workshop on neurogenesis and aging and ADRD that I'll be talking about in a few minutes. I also mentioned to you, we recently have a transposable elements, aging and Alzheimer's disease workshop. We had the cell senescence workshop that resulted in a funding opportunity. We recently had a funding opportunity in glial cell plasticity in aging and AD, and also oscillatory gene expression in aging and AD. Again, trying to look at this interface. And in the next one, just wanted to mention this workshop. It actually happened at the onset of the whole COVID-19 crisis. So we switched very fast from in-person to virtual, and we're able to rescue the workshop. A really tremendous effort by Molly, Brad, Amanda, and Matt. And in this workshop, there was tremendous discussion about really the role of neurogenesis in an adult human brain, regulation of neurogenesis in the aging brain and in Alzheimer's disease, and the functional significant adult neurogenesis in humans, with a lot of new technology being presented, including in vitro models, organoids, neurogenesis, the role of the blood-brain barrier and blood in neurogenesis. And again, this controversy about if in Alzheimer's disease there is a defect on neurogenesis or not, and if it could be targeted therapeutically. And next one, please. Now, another area where we have seen also a lot of growth this year is in new centers. And I just want to be everybody aware of all these new centers that we're funding. This is resources for the community. The Alzheimer's Center is obviously playing a very central role here. And I mentioned before, but I will mention it again, the Center for Alpha-Synuclein Strains in AD and ADRD at Penn with John and Virginia. Just recently, and I will say a few words about this, is a platform for cell type level interrogation for ADRD, Ed Lane at the Allen Institute. We just funded also another center for the next generation proteomics, and I hope to talk to you more about that in another occasion. And also now both Arthur and Lefty have been fused into all FTD for frontotemporal lobe degeneration, and very important programs also connected to the Alzheimer's centers like Diana and BioCard were recently refunded. So really very exciting, large programs serving the community. So, and the next one, please. This is the U19 center that Ed Lane just got funded for single cell transcriptomic and epigenomics. Not only he's going to be looking at this statically, but also we'll try to do dynamic studies. It has a project with the University of Washington, including the neuropathology core of Dirk and Kaiser, another project on single nucleosomics, spatial omics at the Allen Institute. And again, all these data through a coordinating center and the Allen Institute will be made publicly available to everybody. And the next one, some of the objectives are to improve methods and single cell omics, optimize and standardize this technology, define topography, cell type, and profile, and again, try to understand as best as possible the dynamics of this process, and obviously sharing all these data with the community, and a lot of it, of course, coming from neuropath from Alzheimer's centers. And the next one, just to reiterate, the U19, the UPenn, John Trojanowski alpha-synuclein strains in Alzheimer's disease center And this center also will be providing standardized PFFs from human animal models. We'll also be providing expertise as to how to develop these model systems, and again, developing new technology and scientific understanding as to the pathological transmissions of strains of various protein aggregates. And in the next one, this is their form. Please go to their webpage and the resources, how to request their help in this matter. And then the next one, please. And I'm coming to the end. Another area where we have been making a tremendous effort in the past couple of years is in the question of understanding the role of pathogens like both environmental but also microbes in the pathogenesis of Alzheimer's disease. And you remember last time around I presented you a funding opportunity that Matt was setting on infectious etiology of ADIADRD that had tremendous success, and we got a lot of very exciting grants funded, and we're looking forward to that data. Also, we had a little bit before that one on HIV and its role in brain aging and ADRD. And more recently, we funded a microbiome in aging and brain ADRD U19. Again, all these data publicly available. Also part of the consortia with AMP-AD and all the AMP related centers. And of course, now most recently, and you heard from Dr. Hodes, we have NIA had a NOSI on COVID-19. And I just want to say a few words about this. Next one, please. We have all become aware during the course of this pandemic that COP2 not only is altering the lungs, but also is altering a number of other organs. And the brain is probably not immune to these. Many patients develop dizziness, headache, impaired consciousness, ataxia, even epilepsy. We have heard that a lot of them develop hyposmia and loss of taste and other neurological signs. And there is evidence in the literature that other SARS coronavirus like COP1 could be found in the CNS and in the CSF, that coronaviruses could go into the CNS via axonal transport or through the olfactory system. There have been just recently a couple of reports from China on SARS-CoV-2 developing necrotizing encephalitis and meningitis, similar to what has been reported before with MERS and SARS. And we know that other common coronaviruses actually could infect the macrophages and neurons and could actually go into the brain, similar to HIV in macrophages. And there has been in the past association of coronaviruses with neurological diseases like Parkinson's disease and multiple sclerosis. So we think that thinking about coronaviruses and the nervous system and ADIADRD is very important. So in the next slide, please. So in the NOSI that Dr. Hodes mentioned, that is NOTAG 2022, there is a section relevant to the Division of Neuroscience where we call for, these are supplements to your existing aging grants. They could be AD or non-AD, but to study in the context of your grant, and these are against administrative supplements, but could also be revisions if it's out of scope, your application. Studies of neurological and neurocognitive symptoms in COVID-19, especially in the context of ADIADRD, studies of the neuropathology of SARS-CoV-2, and we started to hear about some brains of individuals that have been autopsied that had Alzheimer's and COVID-2, but also, and that means my time is over, development of computational informatic methods So just remember to please include in the box 4B of the application the NOT number, and to remember that these projects are limited to 1 year. And again, these are mostly for administrative supplements, preferable that they are on scope on COVID-19, but on scope with your application. But if they're not scope, we can also review them as a revision. And again, just next one, please. Just to thank you for your attention and remind you some ways to keep communicated. We feel it's very important in a situation like we're living all right now to all stay in communication. Thank you very much.
Thank you very much. That was very great. Nina, I want to introduce Dr. Silverberg, who is the director of the Alzheimer's Disease Research Centers. Nina?
I'm here. Did we want to see if there were questions for Eliezer?
Oh, I apologize. I didn't see any. That's why I didn't.
Okay.
But are there any questions? I don't see any written in unless Cerise found something.
Okay. All right, then I'll go ahead. First, I want to express appreciation for everyone who has kept us informed, given the current COVID crisis, about what's going on with your centers and your participants. We really appreciate you doing everything you can to keep the participants and the staff and your families safe. So please continue to do that and keep us informed of where you are. I also want to very much thank Elizabeth and Maggie for putting together, and also the rest of NACC for putting together this webinar on very short notice. Virtual meeting is not easy to do. And we appreciate everyone's flexibility and understanding of what is needed to put this together. I also want to thank Cerise and Grayson for helping organize everything and helping me with my slides and helping all get everybody all organized. So thank you all for that. There are obviously pros and cons to having this be a virtual meeting. We are able to have 500 people registered, which is more than we would have in person. So we think that's a really great opportunity. And also we were able, because of the COVID situation, we thought it was important to include perspectives from the Neuropath Core and Data Core, which would normally not be at the spring meeting. So I think, you know, we're trying to take advantage and leverage the unfortunate situation to do some good things, we hope. So next slide, please. So just wanted to draw your attention again, in case anyone missed it. There was a notice that went out to the listservs from Cerise, indicating that we have availability of supplements to help with imaging efforts in connection with the SCAN that has now been funded, SCAN project. And those are due May 22nd, and there will be much more discussion of that during the imaging session later today. And I just sent out yesterday an opportunity for centers to apply for an administrative supplement to help with veteran recruitment. And for that one, we're going to ask for a letter of intent, which will be due at the end of May, and then the applications will be due June 12th. And all these administrative supplements, including some of the ones Eliezer mentioned, should go through the PA 18591. Next slide. So again, with the COVID situation, I wanted to really very much thank our clinical measures and diagnosis work group and the cognitive working group for developing the remote assessment battery that you'll be hearing more about soon. That was a tremendous and rushed effort, and we really, really appreciate all of these people's efforts. And special thanks to Bonnie Sachs and Steve Rapp from Wake Forest, who worked with Suzanne Kraft to put together the SOPs and the draft for everyone to see and review before we had this meeting. So thank you to that team. Next slide. And along those lines, we wanted to highlight a few of the many accomplishments that we've had in the recent year or so. These are just a few examples. The remote assessment is the most recent. And in my future slides, you'll be seeing a few of these others. But we consider a lot of these things a success, and many of them have come from the recommendations for the Alzheimer's Centers program. Others are just things that have come up over time. And we can go to the next slide, which is just to acknowledge the paper on late. I know there's some controversy about it. Not everyone agrees, but I think that's science, and we look forward to having that move forward and get more feedback about it and more research on it. And so we consider this one of the many accomplishments of the center's program. Next slide. Uh-oh, okay. So we wanted to be sure that you all were aware, and these are listed on the NACC website. There's a listing of all the different steering committees. The ones shown here, in gray, don't exist yet, but they're in our future plans. We have steering committees for all the cores, and we now have finally a clinical task force, which is a good thing because it helped us deal with the COVID situation. But wanted to make sure that everyone was aware of all the different committees, and we also have interest groups listed on the NACC webpage. Next slide. So for each of the steering committees, for each of the cores, we worked with NACC, NIA, and put together for each committee duties, terms of office, election process, and these are decided in the committees and they can change. But what we're requesting is, I've mentioned this several times at previous meetings, but I want to be sure people are aware and absorbing. We are really increasing what the steering committees are doing from the past. They used to basically just put together the agenda for the in-person meeting that was annual for most of the committees. And now we are having much, much more interaction within the committee using the listservs. And we're asking each committee to put together 1 to 3 major goals during their term. The chair will work with the committee and with NACC and with NIA to put together these goals and make sure that they are accomplished. And they will utilize the figure on the right is from the panel that helped us develop the recommendations for the Alzheimer's centers. And those can be used to form the major goals, but we're not requiring that in any sense. So next slide. One of the other accomplishments that I wanted to draw attention to is the Latino ADC Special Interest Group. It's been really ramping up very recently, and kudos to Yakil and Katya for leading those groups. We have subgroups of that main group, a research working group, which is being led by Yakil and has a website with information about ongoing research studies with a focus on Latinos, list of potential reviewers for papers and grants. The group is also exploring many opportunities for cross-center research collaboration to improve representation of diverse Latinos into our studies. And, you know, this will require a lot of work, but the group has really made a lot of progress. There's also an assessment working group, and their goals are develop preliminary norms for the Spanish UDS-3 and also explore whether the current norms apply to English-speaking Latino participants. So a lot of work there, and they're doing many other things as well. And there's an outreach, caregiver, and central repository working group. And they're working with NIA. We have Holly Massett working, some of you have heard from her, on many materials, develop culturally appropriate outreach and recruitment materials for Latino participants. And I strongly encourage you all to follow the group on Twitter if you have an interest in this area. It's a pretty active group. Next slide. in addition to our accomplishments, we're also looking toward the future. So for 2020, we have many different goals for a lot of different areas. A lot of these, of course, are covered from the panel recommendations that we had and have been presented many times, but also other ideas. And we're happy to work with the different steering committees or any individuals with new and exciting ideas for how we can leverage the center network. Next slide. So I just wanted to make folks aware that we NIA are working with CDC and HRSA and ACL to put together a series of webinars, and the link is there. You can sign up for the upcoming one, which had been planned in advance of COVID, but is timely and appropriate for May, and future ones are listed at that link. Next slide. Another issue that I wanted you all to be aware of, another group I should say, is used to be the Biomarker and Genetic Disclosure Work Group, and is now called ADRD, Advisory Group on Risk Evidence Education for Dementia. We changed it from just a focus on genetic and biomarker to be more open to other areas that we might be interested in, making sure there's some standardization of the disclosure process and information. So the group's aims are, again, to evaluate the science around risk and disclosure to help both NIA and all other researchers guide when and how to provide risk evidence education, and to provide consultation to support ethical and legal risk mitigation. We do have several legal folks in the groups. And I'm not going to read through them, but you can see there are 6 subgroups. The leaders are listed there, and this is a very open group. We've had many people joining over a long period of time. It's open and anyone is welcome to join. Wanted to just report on some of the progress the group has made. There are multiple abstracts and papers and other products that are either done or in process. The group has provided consultation to other groups. They're working on educational seminars, and there was recently funded a grant from members of the group that was supported by efforts of the group. So we're really looking forward to more and more coming from this very large group. Next slide. So this is something I have been trying to do for quite a while. It's finally getting going. I'm very pleased the groups have been put together. We are redoing the Biospecimen Best Practices Guidelines. They were last completed in 2014. And so there are now subgroups put together for each of these areas. There's an addition of a microbiome section now, and it's going to be in future iterations now more of an open and ongoing document rather than a completed product that just sits there for years. So it's going to be kind of more dynamic and, like I said, ongoing updates. Next slide. Oh, and I should just thank all of those many work groups and leaders. The administrators have put together those work groups, and I really appreciate Krista Mulder's effort to organize the whole project. This slide is just to share with you that we did an evaluation thanks to the NIA Office of Portfolio Analysis and Evaluation and Grayson Donnelly, who has put together this slide showing the information that we've gathered to summarize the success. We view it as a success of the Neuropath trainee supplements. The picture on the right comes from the presentations at the October 2017 Fall ADC meeting. That was viewed by pretty much everyone there as a major success. People got to show posters and do presentations and meet with and talk to lots of different folks. one of the major goals of that supplement program was to increase the number of potential leaders for the neuropath cores of the Alzheimer's centers. And we're pleased to say that 8 of the 15 trainees that were funded through that effort have now senior roles in the neuropath core of the centers. And you can see listed there the grant awards that I don't know if we can 100% attribute it to the supplements, but at least have followed on to that work. So we definitely view that as a success. Next slide. And in the same vein, in terms of neuropath, this is leading into, I'm going to be talking a little bit about Neurobiobank. We did a blog post, which is aimed at the researcher community, talking about how we're working, NIA is working with Neurobiobank now, and what our goals are for brain donation. Next slide. So this is a project that has been led by Dirk Keen, and he has been working for a couple years now since he was the steering committee chair of the Neuropath Core Steering Committee. He's been working with me and Neurobiobank folks and the Neuropath Core Steering Committee on a project to collaborate with Neurobiobank. You could see the goals listed there. We wanted to basically make sure that the brain donors' wishes are matched as best as possible to the research needs for brain donation. And so he has organized a pilot with, it was 3, and now Wake Forest will be joining, the University of Washington, Yale, and Pittsburgh, to do a pilot to try to work out how to use the Brain Donor Project and Neurobiobank to funnel sort of the brain donors to the different centers and then make the tissue available through Neurobiobank. Next slide. So we see this as a benefit, hopefully, to both the Neuropath Cores and Neurobiobank. It's promoting collaboration across these different groups. Hopefully it's gonna allow the ability to accept a greater number and also to provide a greater number of appropriate samples to researchers who need them. So the Alzheimer's centers have better characterized samples, but Neurobiobank might have different diversity and different focus. So again, trying to cross-pollinate best practices and improve and harmonize methods across the different groups. So next slide. Just wanted to make sure that you all are updated as far as we can for the future ADRC directors meetings. Obviously, there's a lot up in the air right now. Current plan is for it to be in person, but obviously we'll update everyone in the future as best we can as soon as we know anything different. But this current plan is to continue with Los Angeles in person. Next slide. And final instructions, you received an email that had the subject heading Spring 2020 ADRC webinar final instructions. So that email contains the final agenda. Please be sure to note that there is a PDF in there with ADSP data harmonization initiative that you'll be hearing more about later. And we have evaluation for the spring webinar going on right now. And just want to remind all the speakers, of course, that it's being recorded and this will be archived for others to view that weren't able to join all of us. And next slide. So we have been trying out this new format. For some of us, it's new. For many folks, they've been using Slack and really find it very helpful, reduces the number of emails flying around and organizes topic areas. So we're encouraging everyone to take a look at the email, and in the chat box or Q&A, There should be, and I'm going to look right now to make sure I see it, from Grayson, how to join the ADRC Slack. So I think that's my last slide. And we can take questions for any of the NIA folks.
Thank you, Nina. So just to reiterate that all of these slides and this meeting will be recorded and available on the NACC website. Also, feel free to ask questions using the QA or to raise your hand. There's several of us trying to look and see if there's questions. And then finally, it seems as if the panelists may not be able to use the question answer. So please feel free to unmute yourself, you're a panelist, and ask a question. So I'll take a minute or less, slightly less, to see if there's any questions or if anybody wants to unmute themselves. Okay, so one of the questions is, has there been any discussion at the NIA, NIH about allowing late submissions of applications after the May 1st date that was previously announced? For example,
that's not, it's still, the policy was May 1st and I can send it out. It's still May 1st. So the deadline after May 1st is still the deadline.
Okay, so the deadline has not changed.
Nope.
Okay.
I'll link the notice.
Okay, we have a question for Dr. Hodes. Given that the government is taking on at least 1.5 trillion in unanticipated spending, do you have thoughts on the prospects for grants that would be eligible for funding after October 1st, 2020?
Well, of course, of that very large amount of money, it's only a fraction, though a significant one, has come to NIH. To date, that funding has been targeted for COVID research. And as you know, there are a number of institutes are working at ways to address this, the most vigorous of which are, of course, defining diagnostics and therapeutics. If it's a more general question about availability of additional funding to institutes for other areas of research, that all remains to be seen. It'll be a part of the appropriations process, which this year, it should be said, our FY21 appropriations process is, of course, delayed because Congress is being delayed in its deliberations. But it's tempting, but not a direct translation to take this huge amount of money being put into re-stimulating the economy and to find cures specifically for the COVID-19 virus with the prospects for NIH support. I think it's reasonably not politically charged to say that we can hope that the way in which NIH supported research in general has been so critical to response to this pandemic is, I hope, going to reinforce what has been the strong bipartisan support of Congress for all areas of NIH research. I hope that will continue and be a positive for all of us. Beyond that, I think I can't answer specifics.
Thank you. We also have a question, Nina, about the members and goals of the biomarker committee.
Yeah, I think I typed a question. I wasn't sure. It depends if you mean the biomarker core steering committee. It has not been formed yet. If you mean the disclosure work group, the goals were listed on the slide and the group is open and we can connect you with the group and provide further information offline.
Okay, I have a follow-up question for Dr. Hodes. The person asking the question about money was not asking about new money, but actually wanting to know about how much of a cut to expect.
How much of a cut in the appropriation to NIA, NIH, or for Alzheimer's research? Was that the question?
I think they're suggesting that because a lot of money has gone elsewhere, will there still be the money? Can we still expect the money that we were expecting before this?
Well, if we're talking about the current year, fiscal year 20, The appropriation stands. There's been no rescission of funds, and we're operating under the full assumption that all of our funding for FY20 will remain, including that's been committed to Alzheimer's related dementias research. No sign at all of any backing off on those commitments. Of course, for FY21, as is the case for every year, it remains to be seen what will happen with our appropriation. And sorry for misunderstanding, but what I was addressing was largely the prospects for those out years.
Okay, we have another question.
You should be able to go, Ron.
Okay. Hi, Richard, Julie, Ron Peterson here. Richard, a question for you is sort of a broad one. Do you think the NIA portfolio is properly positioned at present to address the primary goal of the national plan, that is prevent, treat AD by 2025? Do you think the portfolio is adequately situated right now?
Well, our intent and I hope execution, with the help of all of you in the research community, has been to constantly revisit and revise our plan, our strategic plan, our milestones, our priorities, in order to maximize the probability of achieving that success. Now, specifically, as we all recognize, 2025 is growing closer every day and every year. I think that right now we have the most responsible, informed, with input from all of you, balance of research that looks on the spectrum that we've been portraying here at this meeting, from early discovery on through clinical trials. Now, for us to have an effective intervention, cure, prevention by 2025, that would mean in general that this would be dependent upon studies that were very much now in progress or beginning, understanding the amount of time it takes to identify an effective intervention. So we are pursuing all of those late phase 3 definitive trials that appear to be most promising, warrant funding. We are not, of course, able to presume that any one of those will be one of, or much less the definitive cure, and therefore we have in the line, as you've seen progressively, earlier stage, more diverse targets. So the short answer, Ron, I hope better be that with the help of all of you, we are trying to position ourselves to most effectively increase the probability of achieving that success by that time, but also are carrying out a research plan that will continue to generate new and most promising candidates for 2025 and if necessary thereafter. If there's more specificity to your question, Ron, be happy to talk about it now or in a further conversation.
Okay, thank you. I also have a...
Julie, if I, maybe just to make sure to the previous question about funding for this year, I don't know if it was in the intent or not, but implicit in our commitment to use this year's funding to full effect is the understanding that in some cases, the research, clinical research and basic research has been put on pause. It may be that this year there will be research projects that cannot use optimally all the funds that were awarded this year, but there'll be funds involved in starting up again, and it may have an implication for out-year obligations. And I can only reassure that we welcome the input from all of you to understand these consequences, and we'll do our very best to accommodate it. We do understand this is not going to be business as usual over the course of this whole year, even though the full commitment of funding remains.
Thank you. Any further questions? I wanted to just mention a comment from David Holtzman that I don't know if the NIA is aware that the ANA meeting scheduled to occur right after the fall ADRC is going to be all virtual. So I don't know if that's something, Nina, that you're aware of or not.
Yes, thank you. We are aware, and obviously we have a lot of considerations to take into account, and we will. I think it's, I mean, if I had to guess, we're probably going to end up going virtual, but just as of now, we have not made that decision yet.
And if you fill out the evaluation, we can make the next webinar even better.
Okay, so to stay on time, unless there's further questions, we're going to move on to our ADRC network updates for NACC, NICRAD, and ADSP. And we're going to start with Bud, who's the director of NACC at University of Washington.
Hi, everybody. I'm here in the trees to say hello this morning and give you a little bit of an update. I'll be very brief this morning and just give you a few numbers and a few accomplishments that we've come through recently. So next slide, please. So here's the total numbers, all of these subjects that you have contributed data on and so on. So now we have a relatively large database in the UDS part. This is not including the older retrospective minimum data set. We have over 42,000 persons in there, and then also about 6,000 with neuropathology data. And as you know, that's been the neuropathology data, as Nina pointed out, has led to more investigations on late and a whole raft of other things. So great database, all combined from all the centers and all the hard work that you do. Next slide, please. So we've done a few little things to get started with the response to COVID-19. You'll hear more about these in the afternoon. It's been a lot of the hard work put together by the clinical task force and the subgroups of the clinical task force to derive telephone mechanisms for getting data from subjects or from proxies and so on. And as part of that, we've also provided fillable PDFs in English and Spanish to the centers. And you'll hear more about the neuropsychological test battery and the likely inclusion of the geriatric depression scale along with that, and then possibly some changes in the milestone form that Dr. Eliezer will mention this afternoon also. So we're awaiting all this discussion, and then we'll put together forms to get out to you and to everyone, to all the centers, and so that you can begin using them, and at the same time so that the data course can begin programming, as we will have to begin programming as well. Next slide, please. The shortened Lewy body disease module is about ready to go. It's been a while in putting together and getting it actually available for you to submit data on, and that should be, we'll make an announcement on that next week as exactly how you go about that. The brief data set form for neuropathology data submission has to do with mostly with what Nina mentioned with regard to the neurobiobank, and so it basically is a brief clinical information to go with those other sources of neuropathologic specimens. We also created a system some time ago. For the last several years, we've been accepting MRIs from the centers, and we're also in the process of, or we've updated that to be much more efficient and run from the cloud and so on. So that's ongoing. We're still accepting those, and you'll hear more about some of that and the segue into the SCAN initiative that's just been funded as well. Next slide. We also, or the steering committee also has indicated a addition of a blood pressure standard recommendation about machines or how to take blood pressures to be reported on in the UDS, and that is finally being put together and formalized. We've also been working very diligently with the ABCDS and NIA to develop a Down syndrome module, and that's going very well. We're still a ways off from having it finalized, but it's quite a bit closer. The NACC website, we've been remodeling that, and that new website will be launched soon. It isn't like the first launch failed. We have relaunched there. We didn't really, it isn't really like a rocket that blew up or something. We'll be launching the new site here pretty soon. And it'll make a lot of things a lot easier, a lot easier to find things, and hopefully a much better experience for you. So I think that's about it. Is that the next slide? Oh, here's a picture of the new website, what it sort of looks like, and so we'll have that available to you pretty soon. I think that's about it for me. Thanks.
Thank you, Bud. Our next speaker is Tatiana Foroud, who's the director of NICRAD.
Thanks so much, everybody. I'm also glad to see everybody, whether it's just your name or your picture. So let's go to the next slide. I just wanted to start by reminding everyone in the ADCs that we actually have a collaboration that extends beyond just the Alzheimer's Center. So what we've just listed here are all of the multi-site studies that we have ongoing right now that involve multiple Alzheimer's disease centers that are sending samples to us. It's partly a reminder just of the collaboration extends beyond the ADCs, and to remind people that as they're thinking about new studies, we'd love to work with you. Next slide. So I'm just going to go through initiatives. Next slide. And the first one, and this is one we're going to walk through, and I'll tell you as you to forward it. This is about ensuring that all Alzheimer's Disease Center participants have GWAS. So this I really wish I was in person talking to you about. So if you think about all the Alzheimer's centers, you've been sending us DNA to NICRAD, and if you forward the slide, the path that we've done, and it's been incredibly successful with the ADGC, Alzheimer's Disease Genetics Consortium, is over 22,000 DNAs have been GWAS through rounds 1 through 11. The data is at NIA-GADS, and the data for each center goes back to your center through NACC. If you advance, we've got round 12 that was sent right before everything happened with COVID. So about 1,300, almost 1,400 DNAs went to be GWASed at CHOP through the ADGC, and same thing is going to happen. The data will be at NIA-GADS, and then we'll go for each center back through NACC. And if you forward one more time, there's been this group of about 5,000 right now DNAs from the Alzheimer's centers that did not meet the criteria for the Alzheimer's Disease Genetics Consortium and weren't going to be GWASed. And I know this has been a frustration, so I'm really glad for anyone who has missed this news is those samples are being pulled and will finish being pulled when COVID is over. They're going to be sent to CHOP to be GWASed just like everything else at the ADCs. The data is going to go to NACC, and so it's going to be seamless. Going forward, you should be confident that you'll have GWAS on everybody that you've sent us a sample for from your Alzheimer's Center. And I just hope that's really great news for everybody. Next slide. The other initiative I just wanted to highlight when we're all together here is the Fluid Biomarker Initiative, and we've talked about it before. The request is to receive blood samples. We have funds for 3,000 Alzheimer's participants. Everybody will have plasma and DNA that's participating in this, and then a subset for those centers that can do that, PBMC, serum, and RNA. Key aspects are we're doing everything the same. Any Alzheimer's Center participant can be sent. There's no criteria for a clinical diagnosis. And all of these samples are going to be linked to clinical data at NACC and any future genetic or existing genetic data at NIA-GADS. Big benefits to the centers are that obviously you'll get all the materials from us. There's funding involved that you can receive through NACC, and that for many centers, this reduces the burden of having to store samples locally, which is we're hearing more and more. We'll be reserving a subset of each sample type for you that you can ask at any time to get back, and there won't be a charge. So we see this as a really great way for all of us to work together in terms of development and evaluation of blood-based biomarkers, really one of the core things we need in terms of better treatments. Next slide. So the other big thing I just want to say then is to pair with that is those of you who are familiar with our ADC catalog, now as this growing number of other samples become available, you're going to see them and you can already start to see them in our NICRAD catalog. So people will be able to request these other sample types that are easily linked to NACC data, the UDS, and to NIAGADS and other genetic data. Anyone who's interested in making requests like that, we have that on our website, and it would come in through the NICRAD Biospecimen Review Committee. Next slide. And then coming soon, we've got some new catalogs that we're finishing up. The first is for the 90+ study. That is Claudia K. Wasimir Corrada's study of older individuals. Angie, that's ideas, and you're going to hear more about ideas a little bit later today. So from the first ideas, we have DNA samples from a subset of individuals. We're also now, when we get back after COVID, we'll be distributing the first set of iPSC lines, and also fibroblasts will be coming available, and brain tissue. And there are going to be 2 surveys, well, one survey that's going to ask you about both, that's going to be sitting in that, I think, the Q&A that Cerise is going to put in there, and we'd like feedback from you about needs for iPSCs and brain tissue. Next slide. My last one is just to thank everyone. I'm more shameless maybe than Bud is. We're getting our renewal ready, and many of you have helped us and written us letters of support. And I just wanted to thank you, and hopefully we'll be continuing this. And just, again, thank you all. If you have questions, I've talked fast. All these slides are available, and hopefully there's time for questions after Jerry. Thanks so much, and stay safe, everybody.
Thank you, Tatiana. Yeah, we'll take questions after Jerry, if there's any questions regarding any of these ADRC network updates. Jerry, are you ready?
Yeah, can you hear me?
Yeah, so this is Dr. Schellenberg, Director, Alzheimer's Disease Sequencing Project, University of Pennsylvania.
Great. Good morning. I'm going to talk about the genetic projects that use the ADDC samples, the Alzheimer's Genetic Consortium and Alzheimer's Disease Sequencing Project. Next slide. And our partners, of course, are NACC with Bud Kukull, where the phenotype data are collected, and NICRAD, Tatiana, where biosamples are collected, and then we arrange to have them arrayed genotyped at CHOP and put them in GWAS studies, and then shared through NIAGADS. Next slide. And for sequencing studies, the one difference is that the DNA goes to the Uniform Services University of Health Sciences in Bethesda under Clifton Dalgard, and then all the data goes to GCAD at UPenn, where the variants are called by a common pipeline, which performs joint calling, and then that's used in sequencing studies and shared by NIAGAD. Next slide. Our last big paper was Kukull et al., and the ADCs contributed close to 11,000 samples from this for the study. Next slide. And then we partnered with IGAP for a total of 74,000 samples. Next slide. Next, and the outcome of that was 11 novel loci, a number of pathways that were highlighted, including quite a number of signals from the immune response, but also from tau and APP and a number of other pathways. The importance of this study is that after we published this in 2019, the summary statistics were available through NIAGADS, and 47 papers have now been written using those summary statistics that are not ADDC or not IGAP. So your data is being very well used and shared. Next slide. So as this continues to expand, as Tatiana mentioned, this supports other studies. There's a neuropath phenotype grant that Tom Montine and Gary Beecham have, and early onset AD that Gary Beecham and Christiana Reitz have to look at early onset AD. Next slide. But we're also focusing now on multiple ethnic studies. Brian Kukull just had accepted a paper on African American GWAS, and that was a significant contribution from ADCs. And we're now embarking on a large-scale trans-ethnic study that will use these data. Next slide. And this trans-ethnic we'll use all the array data from all cohorts, all the sequence data from all cohorts. Will be imputed with TopMed R2 panel, and we're going to generate an ADRD-derived imputation panel. And the populations that we'll be looking at are listed below, including a large study from India, which samples all across India. So if anything, that in and of itself is a multi-ethnic panel. Next slide. So the sequence data release, our first release was almost 5,000 whole genomes in 2018. We released the CRAMs and GBCS first, and then the project level VCF follows after that. Takes a little longer to prepare. Next slide. Release 2, which the GBCFs and PVCF, or GBCF and CRAMs were released in November 2020, and this is 20,000. I'm sorry, this is release 1. This shows that the case control samples were contributed by ADC. Next slide. And then release 2 is 20,000 exomes, including samples from the ADCs. This is a very complex data set that, because the exome capture comes from a variety of different protocols, so trying to get this in a usable form has been quite an effort, but the CRAMs and GPCS were released in February, and we should have the project level VCF here in the next month. Next slide. And then release 3 will be 17,000 whole genomes, and this shows the ADC contribution. I think we've got about all the autopsied individuals out of the ADCs are getting whole genome sequence. Next slide. And then there are 2 new initiatives that are going to enhance the genetic studies. There's a phenotype harmonization study grant for FOA, and NACC phenotypes will be an important part of this, and we'll try and harmonize phenotypes from a variety of different cohorts that we're using in our studies. Next slide. And then when we identify a signal in our GWAS studies, it's not necessarily telling us it's the next gene or closest gene. It can be a gene, 2 or 3 genes down the road. So there's a new initiative that Alice Yao is the program officer for, to do functional genomics so that we can resolve which signal is linking to which gene, and this should greatly expand the biology that is available from these studies. Next slide. And these are the people that do here: Case Western Columbia, University of Miami, Penn, NACC, NECRAB, and it's a team effort to do this. Thank you.
Thank you, Jerry. We have a couple questions. We have a question for Bud. Let me see if I can find it here. I believe it's about the milestones. What are the specific COVID-19 items to be included on the milestone form?
Oh, I don't have those directly in front of me, just to reflect whether a delay in a visit might be due to the COVID thing, and those will be discussed later this afternoon. There's a session around I don't know, 3 o'clock Eastern time or something like that.
About more clinical core issues, right?
Yeah.
Okay, thank you, Bud. And then Suzanne Kraft, you have a question?
Yeah, I had a question for Tatiana. We had been talking at one point for those of us participating in the biomarker initiative that, particularly on the CSF portion, that AD biomarkers would be returned back to the center. Is that still the plan?
It's a great question. So as you can tell from the fluid biomarker, it's blood-based, this initial round. And so what the initial funding that we received as a supplement is to collect the samples. I think there's sort of 2 paths toward getting the biomarkers generated on. Let's start the blood-based samples from plasma. I think originally we've talked quite a bit about there potentially maybe being a U19 proposed to do that. I will admit that I think has been delayed for a variety of reasons. Another possibility, though, is for researchers, if we want to have a collective group request potentially all available samples and run on certain platforms. So I would say, and I apologize for that, we don't have it worked out how the biomarker data is going to get generated back off plasma again, because we don't have centralized CSF from the ADCs yet. I don't know if anyone from NIA would want to comment on that. I think there'd be a lot of interest potentially in receiving requests that might be able to generate that from plasma. I think the whole advantage here is to get it on a single platform. Okay. Can I shamelessly do one other thing, Julie, is if anyone's trying to complete the surveys, only because I just tried, you have to copy the link, you can't click on it. And I just want to encourage people to click on, shamelessly, that the link for the 2 NICRAD questions that we have, but just make sure people know how they have to do that.
Thank you, Tatiana. We have a question from Charlie DeCarli, but I think Cerise, you're going to have to say what the question is.
So Charlie's question is for Jerry. He said he noticed that there's no Hispanic individuals in the work. Are there plans to include Hispanic individuals given the growing diversity of the ADRC program cohorts?
Actually, there's a variety of Hispanic cohorts in our GWAS study. Some of them are Caribbean Hispanic from Richard Mayo. We're working with the TARC group, Texas Hispanic. We're working with 2 groups in Mexico that have Hispanic people. And then there's auxiliary studies going on in Peru, Puerto Rico, of course, Dominican Republic, and another number of other South American countries. And then we're collaborating with Agustin Ruiz in Spain, who has a very large Spanish Hispanic cohort doing a all Hispanic GWAS. So I'm sorry I didn't mention that, but we actually have a pretty heavy emphasis on Hispanic. We emphasize every ethnic group that we can get our hands on, but actually Caucasians and Hispanics are the largest group that we can actually access.
Okay, thank you, Jerry. So we're running a couple minutes over, so but I'm glad we got a lot of questions in and answered. Let's move forward with an update on the IDEAS trial. This will be from Gil Rabinovici at the University of California, San Francisco. Welcome.
Thanks, Julie. It's really a pleasure to be here. I can start my slides. There we are. Next slide, please. So IDEAS is supported by a public-private partnership. The PET scans in IDEAS are covered by Medicare as part of a program called Coverage with Evidence Development, which allows them to assess the clinical usefulness of new technologies. The study is also funded by the Alzheimer's Association, by 3 industry partners, AVID, GE Healthcare, and Life Molecular Imaging, and is operated by the American College of Radiology. Next slide, please. So what I thought I'd do today is start with a brief overview of the design and objectives of IDEAS. I'll give a very high-level overview of our AIM-1 results, where we looked at the relationship between amyloid PET scans and patient management. This was published last year. I'll give an update on the timeline of our AIM-2 analysis, where we're evaluating the associations between amyloid PET scans and health outcomes. I'll give an update on a few secondary analyses that are ongoing in the study. Next slide, please. And I'm very excited to announce publicly for the first time to this community that as of last week, CMS has approved a follow-up new IDEAS study. This study will allow us to really improve the diversity of patients that are included in the IDEAS program, and also to establish a biorepository that will collect DNA and plasma from IDEAS participants. So I'll conclude just by describing the design and aims of the new study. Next slide, please. So IDEAS was really established in response to a call from CMS to develop studies that would allow them to evaluate the clinical utility of amyloid PET and make decisions about reimbursement. It's a single-arm, multi-site longitudinal study where we assess the clinical utility of amyloid PET in about 18,300 Medicare beneficiaries who have MCI or dementia and meet amyloid PET appropriate use criteria that were published by Keith Johnson and colleagues. And I'll review those criteria as we discuss the inclusion and exclusion. Participants are referred by dementia specialists for PET scans with one of 3 FDA-approved amyloid PET ligands, and the study has 2 aims. The first is to evaluate the impact of the scan on patient management at 3 months following the scan, and the second aim is to assess the impact on major medical outcomes 12 months following the scan. And really the premise of this study is that in these diagnostically uncertain cases that meet appropriate use criteria, amyloid PET is going to lead to significant changes in patient management, and these will then translate into improved outcomes. Next slide. So here is a partial list of inclusion-exclusion criteria. To be in IDEAS, individuals had to be Medicare beneficiaries over age 65, English or Spanish speaking for consent, and they had to meet these appropriate use criteria, which basically meant they had to have objectively confirmed cognitive impairment, so MCI or dementia. The study did not include cognitively unimpaired individuals. The cause of cognitive impairment had to be uncertain after a comprehensive assessment by a dementia specialist that included the standard lab tests as well as structural imaging with CT or MRI. And then finally, knowledge of amyloid status had to be expected to change management. The primary exclusion were people who already knew their amyloid status from previous studies, people who were previously enrolled in an anti-amyloid clinical trial, where interpretation of the PET result would be complicated, individuals in whom the knowledge of amyloid status was expected to cause psychological harm, people who had a short life expectancy, or were already in a skilled nursing facility. Next slide. So this is the basic flow in the study. Patients are assessed by a dementia specialist at a pre-PET visit, and the specialist then documented their diagnosis and care plan, assuming this was the end of the diagnostic route, and assuming they had no access to amyloid PET. We then did a scan with an FDA-approved ligand. These scans were interpreted locally. The results were communicated to patients. The dementia specialist made recommended changes to the care plan, and then the patients were seen again 90 days after the PET scan. And at this point, the dementia specialist documented the care plan now implementing amyloid PET results. And again, in the first aim, what we were looking for was a significant change in the care plan between the pre-PET and the post-PET visit. And then we're continue to follow Medicare claims in these individuals for 12 months. And the second aim is to assess whether major health outcomes like hospitalizations, emergency room visits, total aggregate utilization were reduced by at least 10% in the study cohort compared to a matched cohort of Medicare beneficiaries who do not have amyloid PET. Next slide. So it's probably refreshing to look at the map without looking at COVID cases. This is a map of IDEAS sites, dementia clinics, PET facilities, and radiopharmaceutical suppliers. Overall, we were able to engage about 600 dementia practices. Certainly a lot of the ADRCs participated, but we're especially proud of the fact that 80% of our sites were actually private or group practices, so sites that don't typically engage in AD research. We reached almost 1,000 dementia specialists. Imaging scans were conducted at 350 PET facilities, 730 radiologists participated. We completed recruitment over 2 years. The median age in the study was 75. About 60% of patients had MCI and 40% had dementia. And here you see the rates of amyloid positivity, 55% in MCI, 70% in dementia. Next slide, please. So last year, we published in JAMA the results of our first aim. Next slide. In this aim, we were hoping to identify at least a 30% change in the management plan between the post-PET and the pre-PET visit in a composite endpoint that included the use of AD drugs, non-AD drugs, as well as counseling and referrals to social services. And as you can see, the effect that we actually measured was twice of what we anticipated. So these elements of the care plan changed in 60% of patients with MCI and almost 64% of patients with dementia. Next slide. Here you can see the change in the components of this composite. The most common change was in the use of AD drugs in about 45% of patients, and then about half those rates in the use of non-AD drugs and counseling. We saw changes in diagnosis in a large number of patients in the study. In 25% of patients, the diagnosis changed from AD to a non-AD dementia. In about 10% from a non-AD dementia to AD. And this was, I think, a very striking statistic for me. 71% of patients who had a negative amyloid PET scan in the study actually carried a pre-PET diagnosis of Alzheimer's disease. And so without access to amyloid PET, these patients would have continued to be diagnosed and treated as if they had AD. Next slide, please. Medicare has made it very clear that they want to see an effect also on health outcomes, and this is aim 2 of the study. Here, our primary endpoint is to see if amyloid PET is associated with at least a 10% relative reduction in hospital admissions and ER visits over 12 months in the IDEAS cohort compared to a matched cohort of patients who did not have amyloid PET. We are also looking at a number of secondary endpoints, preventable hospitalizations, aggregate resource utilization, and also looking at these associations in subgroups, individuals who are amyloid PET positive versus those who are negative, people who have MCI versus dementia. And so we finally have the complete data set and are completing analysis, and we will release these results really in the next few months, certainly in the summer of 2020. Next slide, please. I think I'll skip this one. Next slide, please. So because this is a practice-based study, this allows us a little bit of a window into healthcare disparities related to dementia care in underrepresented minorities. And so the most obvious disparity is the low participation. Overall, only 10% of IDEAS participants came from an underrepresented minority, about 5% Hispanics, 3.5% Black, and 2% Asians. However, even though the percentages are small, this is still 5% of 20,000. And so the overall numbers allow us to do at least some preliminary comparisons with white participants. And you can see that compared to whites, all minorities and IDEAS were more likely to have dementia at study entry. For example, 52% of Blacks, 56% of Hispanics had dementia compared to 37% of whites. Minorities were more likely to be enrolled in a Medicare Advantage plan. They had a higher prevalence of diabetes, higher prevalence of hypertension in Blacks, and a lower prevalence of a family history of dementia. Next slide, please. Here are the results of amyloid PET positivity in minorities and IDEAS compared to whites. Here you're seeing dementia in red and MCI in blue. So you can actually see that the higher rates of amyloid PET positivity for each clinical stage were in whites, and all minorities had lower rates of amyloid PET positivity. And I think this converges very nicely with neuropathological data out of UC Davis and other centers that show a higher contribution of other causes of cognitive impairment, for example, vascular causes in minority participants compared to whites. Next slide, please. By design, IDEAS participants were referred to a number of add-on studies to gather additional data. This included ANGI, that Tatiana mentioned, a genetic study, and the Brain Health Registry, led by Mike Weiner. These are funded by the Alzheimer's Association. NIA funded a caregiver-oriented study where dyads of patients and caregivers and IDEAS are being interviewed, and C2N Diagnostics is looking at plasma A-beta in IDEAS participants. So look forward to data really in the coming months to years from these add-on studies. Next slide, please. New IDEAS, as I said, is approved by CMS. This study will really allow us to improve the diversity of IDEAS participants. We are going to recruit an additional 7,000 Medicare beneficiaries, including at least 2,000 African Americans and at least 2,000 Latino Hispanics. The first IDEAS study really focused on atypical presentations, but in this study we'll also be able to enroll participants who have typical amnestic presentations of Alzheimer's disease. We'll be able to additionally enroll Medicare beneficiaries who have early onset dementia, and I think really importantly, a biorepository that will collect DNA and plasma on these participants, and it'll be very interesting to compare genetic risk factors and plasma-based biomarkers in this biorepository compared to the one that's being established at NICRAD. And so we hope to launch this study in late summer or early fall, COVID-19 permitting, and anticipate recruitment over 3 years. Next slide, please. These are the study aims. We are making a little bit of a change in how we're looking at claims. So we're going to be comparing across this diverse cohort claims in amyloid PET positive versus amyloid PET negative individuals is our first aim. And then in aim 2, we're going to be looking at the association.
Where the hell is it?
Sorry, Gil.
Yeah. Oh, sorry, Cerise. Okay. We're going to be looking at the associations of amyloid PET findings and health outcomes in different ethnic and racial minority groups. In aim 3, we're going to compare these outcomes in patients with typical and atypical forms of Alzheimer's disease. Next slide. Now, we're really happy to add some amazing people to our IDEAS leadership team, including Drs. Consuelo Wilkins and Peggy Dilworth-Anderson, who will be leading our minority recruitment efforts, and Drs. Sid O'Brien and Rob Rissman, who will be leading the biorepository. Next slide. So this is just a final slide. IDEAS study so far has provided, I think, the strongest phase 4 data to date to support the clinical impact of amyloid PET on patient management. Look forward to our aim 2 results, looking at health outcomes, and this will really inform coverage reconsideration sometime this summer. New IDEAS is coming soon to a PET scanner near you. And I also really wanted to emphasize that IDEAS data should be considered a resource to the research community, ADRC community, and beyond. We are currently accepting data sharing requests and reviewing them through a research and publications committee, but we also hope to upload IDEAS data and images to the Global Alzheimer's Association Interactive Network. Next slide. So really just wanted to thank the very many people who have made this study possible, especially Maria Carrillo from the Alzheimer's Association, the fellow study co-chairs, our partners in industry and CMS. This is a photo in less socially distanced days about 5 years ago from our first IDEAS steering committee. And so we hope to continue this program through the new IDEAS study. Thank you very much.
Thank you. Thank you so much, Gil. That was great. We have a question, as you think about other questions for Gil, we have a question or hand raised from Doug Galesko.
Through text as well. Do you have any information on the accuracy of community diagnoses of amyloid positivity or negativity? And this is something you're going to look at.
Yeah, so we do plan to compare the visual reads that were done in the community to both quantification of the amyloid PET scans. We have about 15,000 scans archived from the first study, as well as some expert visual reads. So the design of that study is in the works, and I think it's going to be very important, especially if these PET scans are going to be used more broadly for treatment eligibility if we do have a drug that relies on a positive amyloid biomarker. So absolutely, Doug, we are planning to do that.
Right. And so as pertinent to that, will you also be able to look at some aspect of either the experience of the community readers or the number of scans they read in relation to accuracy?
Yes, we also have information about years of experience of the different raters. Also, we also have that information about the dementia specialists, so we can evaluate how their experience related to their prediction of the amyloid PET results. So we are going to be able to look at that. This is really a goldmine of data, and there are a lot of secondary analysis that will be very interesting and inform us about how dementia care is occurring in the real world.
Okay, thank you, Gil. We have a couple comments and another question. We have a couple that say that you gave an excellent presentation, Gil. That was from Charlie DeCarli. We have a comment, very nice study, just a comment that lower rates of AD in Blacks and other minorities might be related in part to enrollment factors.
Yeah, so we're going to be looking at that. That's a really excellent point. And so Drs. Consuelo Wilkins and Peggy Dilworth-Anderson are conducting a deep dive into the minority participants. And one of the things we're going to do is identify white participants that are matched in terms of their enrollment factors, comorbidities, and evaluate whether the PET results are related to those factors or independent of them. Excellent point.
Great, thank you. We have another question. The IDEA study can be of great benefit to minority senior adults who are not seen in neurology practices. Are there geriatric medicine or family medicine clinics, whether through academia or federally funded healthcare centers, to be included in this next iteration of the IDEA study?
Yeah, great question. So first of all, dementia specialists don't need to be neurologists. So we actually do include geriatric medicine and psychiatry as part of dementia specialists. The point of referral and new ideas will still be the specialty clinic. However, we are going to make specific efforts targeting both patients and families as well as primary care providers in the communities to make them aware of new ideas, and that we hope that this will facilitate referral and better access of minority populations to the specialists. So we are not yet allowing referrals from primary care for a variety of reasons that we can discuss online, but we are going to be making those communities that are aware of the study and hope that that will facilitate referrals.
Another question, can you comment on enrollment recruitment strategies across racial ethnic groups that can inform the question of whether enrollment bias is contributing? And it sounds like you've kind of already answered that you're still looking at it.
Yeah, and I have to say, you know, and this is a mea culpa, you know, the first IDEA study was really all comers, so we didn't have a specific strategy to improve the diversity of the study. And in this study, we have considerable resources and real experts in the field of recruitment science, making our recruitment strategy. And I can't really, it would take me, at least 10 minutes to describe all the different strategies that we're going to employ, but I'd be happy to come back and report on that or discuss that offline. But it's going to be a really concentrated effort, perhaps unprecedented in the field, to engage minorities in this study.
Another question, are there data collected and ideas on potential vascular risk factors or vascular brain injury? If not, will the new IDEA study plan to collect it?
Yes, both studies. So in the first IDEAs, we had specialists fill out very detailed case report forms where they documented the presence of vascular risk factors, cerebrovascular disease, previous history of strokes, white matter, et cetera. And so we're starting, I had to skip that slide for time, but we are digging into those factors and many others in terms of how they relate to amyloid PET positivity. And we are planning to include all of this in the new study. However, if people, we're still really in the early development phase of designing the case report forms for new ideas. So if folks have specific things that they want to recommend, please feel free to email me. This is a great opportunity to get broader input as we design these forms.
Okay, I think I'll take some more questions because I think we're